Thank you, Neha, and thank you, Professor Quidant, for the very engaging talk. We have a lot of questions pouring in, and before proceeding with that, I would like to take this opportunity to thank my co-moderator, Dr. Vidika Punjani, who has been compiling these questions for us. And now, without further ado, let me jump right into our first question. How does digital holography improve the sensitivity or specificity of biosensing compared to traditional optical methods?
Thank you for this question. So I would say the strength of this digital holography is dual. So the first advantage is that you're able to resolve each of the binding events instead of integrating over a large, let's say, amount of analyte that would modify your sensor. And the second one is, in fact, this is very sensitive. So not only to single binding events, but also to analytes that would be very tiny. And so doing single protein sensing with a conventional sensor, so some of them enable it, but this is actually very, very challenging. And here you have it really like you can do like high throughput single molecule detection, friends.
Thank you. Thank you for that answer. We have another question from the audience. Is it possible to study these complex functions or interactions comparably with this technique in simple heterogeneous catalytic systems like inorganic materials-based nanoenzymes?
Interesting. I haven't thought about that. So, so of course, the example I was showing is proper to a bioassay. But if you would go to something that is a different kind of matrix where you have a very specific kind of, let's say, landing of a given object, particle slash molecule on the surface, you should be able to extract relevant information for your system. So I would say like maybe
site-specific binding on surfaces.
Exactly. You need that to happen, and you need your system to be transparent somehow. So otherwise, this is complicated. So you need, if your sample is non-transparent to the light that is used to interrogate, this is maybe where you may reach complexity.
True, true. Yes. We have another question. What is the resolution limit of IceCat microscopy, and how do you ensure non-interference from autofluorescence of samples?
Good question. So resolution is an optical resolution. So this is conditioned somehow by the numerical aperture, the objective that's going to be used in the microscope. So typically, if you push to 1.2, 1.3 for numerical aperture, you would be able to have a resolution in the order of a couple of hundreds of nanometer resolution. So what I meant with that is that this is not a super resolution technique. So, you know, it's not like STED or STORM where you kind of beat the diffraction limit. Here, you are still limited by the limit of diffraction. The second thing is actually a very important one, is that this is a bit related to what I was trying to explain, that you need a very clean surface. You need, for instance, to minimize the roughness at the surface of your chip, because otherwise this will contribute to background. Anything that is, let's say, generating scattering around your analyte will affect the signal-to-noise ratio of your measurement. And another thing that is a little bit more technical, but the coherence of your light is also complicated, let's say, can generate some complication because it will generate speckle. And speckle is kind of also a kind of coherent background that's going to, let's say, reduce the contrast of your analyte on the image. So there are a few things where you need to be very careful with.
Maybe on a related topic, I will ask this question. What has been the biggest technical challenge in developing this platform, and how did you overcome it?
That's also a very good question. So I would say, so you have seen the microscope, at least when represented as a sketch. So of course, I'm the one in the office. All these smart people have been mentioning are the ones doing the work in the lab. So without minimizing the effort, the setup per se, when you look at it, is a quite simple one. This is far from being a complex optical setup. However, in order to extract the right level of sensitivity, and in particular to extract the signal from the background noise, there's a lot of steps, and these steps are on the hardware, but they are also on the software. So digital holography and IceCAD are kind of very much demanding in terms of data analysis in order to kind of process the big amount of data you're going to extract, and also to extract small signal from a background. So these would be the main challenges. Yeah.
Thank you. Another question on a related topic was, what are the computational requirements for reconstructing this holographic data? Since we touched upon that just now.
So, directly related, so they are quite demanding. So, and so in this specific case, we are detecting in at the surface, but they are off-axis holographic technique that actually detecting throughout the volume. And so so this kind of a volumetric NTA, for those of you who are familiar with that. And so what is the problem there is that you end up with a huge amount of data. And so you need to be able to handle that. And there is actually, we are getting into really the field of computer vision. You need really to do like a very heavy data management.
Thank you, Professor Quidant. Next question that we have is, this is pertaining to the last part of your talk. So what kind of applications do you envision to target by mapping enantiomeric excess, and what kinds of biological questions that can be addressed, and how, now that we can visualize chirality and enantiomeric excess at the single particle level?
Yeah, that's also a very good question. So I would say we aim at going step by step. The first interest we see, I'm sure you're not, you are familiar with the fact that, for instance, there are many aspects lately of material sciences, and also, for instance, photocatalysis that are involving chirality, where, for instance, the catalyst is actually chiral in one way. And so before going to the biology level, I think, for instance, being able to characterize these kind of nanoparticles that have a structural chirality and are used, for instance, to catalyze a chemical reaction, we would extract a lot of information. They also, for instance, in the quantum optics community, people are very interested in material that would generate single photons, but with a certain spin. So possibility also to characterize this kind of emitter using this kind of microscope. And from a more biological standpoint, so now we are trying to talk a bit to potential end users and to identify what are the right questions. But so, of course, molecules, being able to discriminate, for instance, in the synthesis, what is the ratio of enantiomer? So it would be, of course, very relevant. But maybe at a larger scale. So for instance, possibility of identifying some chirality change on tissues. So there are some studies that are showing some diseases may induce this kind of modification structurally. So there is a broad range of possible applications, but we are honestly trying to narrow down a little bit what are the main questions right now.
Sure. Thank you. That was very insightful. On a related topic, are there any ongoing efforts to miniaturize or commercialize portable biosensing devices using these methods?
Yes. Yes, there are efforts in this direction. So there have been already a few initiatives that are not going from our labs, that are coming from the lab of some of the colleagues I was mentioning before, particularly the group of Philippe Kokura. So there's a startup that is kind of using digital holography for monitoring basically the molecular weight of individual proteins or small objects. And so we are thinking that, possibility of using that for actual bioassays. So what I've been discussing the first part could be an interesting direction. And there is room for devices here. This is not a technique that requires a huge amount of space. And so there's possibility for integration indeed. Yeah.
Thank you. We have another curious interdisciplinary question, I would say. What role can physics play in advancing the biooptical instrumentation and quantitative imaging techniques?
It's a very good question. And And so as it was said before, some of this is by training. And so I think there are many things where physicists, when they are a bit more open to talk with chemists, biologists, and etc., actually can really bring their own contribution. So in that case, for instance, really the physics aspects of detection of very tiny signals out of a background, what I've been insisting on several times, is actually something that is here that is crucial. So having the ability to understand how you extract a very tiny signal from this background is something that we directly use from a physics background, and we apply to the specific more biochem, let's say, application. So I would say there are many aspects. So there are many aspects where physics can contribute. The right thing is actually to connect dots. No, how do you connect a know-how that probably may have nothing to do at that point, for instance, from quantum optics, and how do you use that to make it useful for chemistry or for biochemistry, for instance?
Thank you. Another related question, I would say, do you envision this technology being used for early stage disease detection like cancer or neurodegenerative disorders where EVs or protein biomarkers are key?
Yes. So we always have to be careful with, let's say, the claims on this specific application. So the application of doing diagnostic, because I'm sure everybody knows in the audience that a reliable, let's say, early stage detection, for instance, like for disease like cancer, they require actually many analytes. So it's not, with a couple of them that you're going to extract a solid conclusion about. So establishing a diagnostic is a big thing. It's a big. You really need to be backed up by a lot of information. And so the question is whether the platform can indeed really provide all this information that is necessary for medical doctors to actually reach a robust diagnostic. This is why I'm not qualified for that. But what I'm trying to say is that we as technology developers need to try to meet the needs of the end users, in that case for diagnostic purposes, and to see whether this is something that where we can reach what is needed. And I would say this is too early right now to say.
Okay. Next question would be, are there any examples where holographic biosensing has led to breakthrough in personalized medicine? As you had mentioned initially, one of the potential applications could be personalized medicine.
Not that I know, not yet. But hopefully soon.
Yeah. Also, coming back to the questions about the data analysis part, so is there any way we could integrate AI or machine learning platforms to help with this?
Yeah, definitely. So there are already some works along those lines, and so for instance, AI can be extremely useful. I think that's been demonstrated by one of the work I was mentioning, so that somehow pushed the limit of detection to 10 kilodalton for single protein. So the possibility, again, I'm repeating myself, but how to extract this very tiny signal from the background. So AI can be useful for that. It can be useful also when you extract a lot of information, let's say from a bioassay, and you want to reach out a robust conclusion. So in the way you're going to interpret the data, and so AI could be of course also very useful here. So yes, I think we are in an era where AI for sure will contribute in many different aspects into this field of biosensing for sure. Yeah.
Thank you. We still have quite a few questions pending, but in the interest of time, we will close this now. And before departing, Professor Quidant, would you like to share with us any concluding remarks for all the young researchers here in our audience?
So maybe what I can do is, if I get out from me, share what I can do. Can you see my slide again?
Yes.
Yes. So is maybe tell you very quickly about our journal, ACS Photonics. And so, so I think what I've shown here today is kind of an illustration of the kind of, let's say, science we like to publish. So we, of course, focused on advances in photonics. Photonics is an enabling technologies nowadays. And so ACS Photonics is kind of this interdisciplinary forum where we bring all the communities actually working on photonics and contributing to photonics. And they can be chemists, physicists, biologists, engineers. And so we have a journal that brings this community together with, I would say, quite strong points. We have a very reasonable impact factor of 6.7, so newly from 2024. And so we try to be very fast as well in the way we process the submissions we have, with an average time to first decision of less than 1 week, and to an average time to an acceptance of the paper of 12.9 weeks. So I really encourage you, anyone working in the photonics community in one way or another, to check the journal and to see whether your next work could be a good fit for the journal.
Thank you. Thank you, Professor Quidant.